Good morning, all of you. Today, we are here for the National E-Seminar on Novel Approaches in Research Methodology, organized by Department of Botany, Research, Innovation and Extension Cell, and IQAC of J.A.T Arts, Science and Commerce College Forum in Malegaon. For that, we have our principal, Honorable Dr. Mohammad Harun Mohammad Ramzan sir, academic and administrative coordinator, Dr. Salma madam, IQAC coordinator, Professor Munawar sir, Dr. Ismail sir, Professor Ataur Rahman sir, Professor Navid sir, and all the teaching staff from the Department of Botany. So very warm welcome to all of you for the inaugural session of today's e-seminar. So for starting the program, we have a president for our program. So may I request Dr. Muhammad Ismail sir to nominate the name of the president and second the same.
Thank you, sir. I feel honored to be here for the nomination of the name of the president of today's function. And he is the head of J.A.T Arts, Science and Commerce College for Women, Malegaon. He is Honorable Principal Dr. Ansari Muhammad Harun Muhammad Ramzan sir. With due respect, I propose his name as the president of today's function and second the same wholeheartedly. And I seek his kind permission to proceed the program further. Thank you, sir.
Thank you, Ismail sir, and thank you, President sir, for giving permission to start the program. Now, may I call upon Professor Munawar sir for delivering the objectives and the need for research in a higher education and in the field of research. So, please, sir, give the objectives of the research methodology.
President of today's program, Dr. Ansari Muhammad Harun Muhammad Ramzan sir, academic and administrative coordinator, Dr. Salma Abdul ma'am, NAC and IQAC coordinators, organizer of this prestigious program, Dr. Pathan Yahya Khan, and his faculty members of the department and teaching assistants and their students and all the participants. Good morning. I cordially welcome to all of you. Today's program is undoubtedly a quality-oriented program because research has become an integral part of higher education. In order to nurture research culture in the college, we have been conducting research methodology programs every year without fail. Research is the sole responsible factor for the generation of new knowledge, but it must be done with proper interest and methodology. With these aims and objectives, our college, J.A.T Arts, Science and Commerce College, is organizing this quality program, and I extend my warm welcome and thank to both the resource persons for being the part of this quality initiative of ours. In the end, I hope that this program would help all the faculty, students, and participants create a genuine interest in doing research and in turn create research environment in the college. Wish you a good luck. Thank you all.
Thank you, sir. For this seminar, we have two resource persons, Dr. Syed Abrar Ahmed sir from Aurangabad and Dr. Azarian Shakespeare sir from University of Johannesburg, South Africa. Now, I may call upon our academic and administrative coordinator, Dr. Salma ma'am, to give the chief guest speech.
Assalamu alaikum. A very good morning to all of you. Honorable President of today's webinar, Honorable Principal Dr. Ansari Muhammad Harun Muhammad Ramzan sir, Honorable IQAC Coordinator Professor Munawwar sir, Honorable Organizer or Coordinator of this national webinar, Professor Dr. Pathan Yahya Khan sir, Honorable Dr. Muhammad Ismail sir from Zoology Department, Honorable Dr. Mumin Adil sir from Chemistry Department, Honorable Professor Naveed Akhtar sir from Computer Science, Honorable Professor Ataur Rahman Noori sir from Urdu Persian Department, Honorable Mr. Momin Abdul Hasib sir, and dear teaching associates and dear participants, we are here for a national webinar on the very important topic, very innovative topic, that is the novel approaches in the research methodology. As Professor Munawwar sir shared about the aims and objectives behind organizing this activity, I forward his talk that this topic we selected due to the National Education Policy, that is NEP 2020, in which there is focus on research culture maintenance in higher educational institutions, keeping in view the spirit of new education policy. We are organizing this activity here on the platform of research, innovation and extension activity along with Department of Botany in association with IQAC. We are organizing this webinar. For this, there is a team who struggled hard for giving a reality, for making reality this event. There is a team. I appreciate their efforts, congratulate them, the entire team. On behalf of J.A.T management, especially Honorable Chairman Alhaj Ansari Nehal Ahmad Sahib, we welcome both the intellectuals who are here connected with us for this webinar on the very innovative topic, that is novel approaches in research methodology. Both are experts, eminent personalities, and within a few minutes we will discuss with them. We will
Good morning to everyone, all the dignitaries, participants and students. On behalf of J.A.T management, behalf of J.A.T Arts Science Commerce College, principal, staff member, and students, I warmly welcome to all of you. And here I am the principal of this college, and I appreciate the whole team of this webinar. And there is very interesting topic today, that novel approaches in research methodology. And we know that today it is very interesting topic and subject in the University Grant Commission, national policy, and in higher education it is very compulsory now to do research. So it is our duty, all the faculty, that we initiate the research methodology in our students, then we can uplift our nation. Now I am giving my best wishes to all of you, and here I conclude my speech. Thank you to all.
Thank you, sir, for your best wishes and the presidential speech that are guiding us to organize such type of seminars, workshop, and conferences. Now, I would like to propose vote of thanks. First of all, I would like to thank our honorable chairman of our J.A.T Trust, Honorable Alhaj Nihal Ahmad Mohammad Harun Ansari Sahib, for its consistent encouragement and having such a great inspiration for us to organize such type of activities in our college. Then after the management of J.A.T Trust, we are also thankful to them for giving us opportunity for organizing such type of program. Then after, I would like to thank our principal for consistent guidance, and in every program he had asked to proceed the program. Thank you, sir, for your consistent guidance. Then after, I would like to thank our academic and administrative coordinator, Dr. Salma ma'am, the IQAC coordinator, Professor Munawar sir, all the teaching staff from the J.A.T Senior College, the technical staff, Naveen sir, Ataur Rahman sir, Hasib bhai, and all the teaching assistants and non-teaching staff from our department, and all the participants that they have consistently here for our encouragement to start the program. Now we will move towards our technical session first. For that, we have a resource person, Dr. Abrar Ahmad sir, Syed Abrar Ahmad sir. Sir is having PhD from Swami Ramanand Tirth Marathwada University on analysis of secondary metabolites by tissue culture technique in 2010. From 2010 to 2016, he had served as assistant professor in Maharashtra College of Arts, Science and Commerce, Mumbai. In 2016, he had been selected through Maharashtra Public Service Commission as an assistant professor and having the post at Government Science College, Gadchiroli. In Gadchiroli, he had done lots of research work, and when he was working in Gadchiroli, he had studied one species of the orchid that was firstly reported from the Maharashtra state, and recently it has been published in the Journal of Threatened Taxa, and it was appreciated by not only in the Maharashtra as the India level. So we have a great resource person that now sir is working in Aurangabad, and he had transferred in 2020-21 in Aurangabad Government Science College. On his credit, there are 15 research papers in reputed national and international journals. Sir is very kind, and on a single telephonic call, he had accepted our invitation for delivering his expertise in the field of research. So welcome, sir. We are very thankful to you and welcome to you, sir. Now the session is handed over to Dr. Abrar Ahmad, sir.
Okay. Well, thank you very much, Dr. Yahya Khan, sir. Am I audible?
Yes, sir, you are audible.
Yes. First of all, I would like to express my gratitude and to Chairman of J.A.T Education Society, Honorable Alhaj Nihal Ansari Ahmad Mohammad Ansari Saab, then Honorable Principal Dr. Mohammad Harun sir, Academic Coordinator Salima ma'am, then IQAC Coordinator Munawar sir, and all the faculty members, participants. Thank you very much for giving this opportunity to deliver a lecture on this a wonderful topic, that's novel approaches in the research methodology. In this time, everyone is knowing research is become an integral part of the academic growth, and various research scholars, the researchers, they are working hard for finding out the novel things. But in keeping in view that this research methodology, the most of the researchers, they are quite blind. They don't know how to select the topic and how to go for the completion of this research work. So if you look at the research methodology, it's such a vast subject. So it is quite difficult to complete the entire subject within this stipulated time. So I'm going to deal today with the reference management softwares. We are knowing that. So when we go for the publication, the various research papers been published in different journals, we are knowing that every journal is having their own style of referencing. And sometimes what happens, we used to communicate our paper to the journal, but due to non-meeting their aims and objectives, most of the time the papers get rejected. At that moment, these researchers get frustrated. So to overcome on this problem, the referencing is one of the Herculean and the very tedious job, because what happens when you try to do the referencing, so there is a chances to miss sometimes the volume of the journal, or sometimes the page number, or anything that means. So if you try to do it manually, it takes much time, and we need to do it very meticulously. So in this regard, thanks to the technology, a lot of people, they have taken efforts who made it available for the researchers, so that we need not to worry about the referencing. So there are so many different online reference softwares are available, like the EndNote, Mendeley, Zotero, and LaTeX, right? So out of these, today we are going to discuss about this Zotero. Well, the reference manager softwares or the tools. So these are the certain softwares, the popular softwares. This are Mendeley and the Zotero. So these are the wonderful softwares which has been widely used by the researchers for a proper referencing. And nowadays, in case of this Mendeley and the Zotero, more than 10,000 different styles of referencing is available. So can you imagine, can we able to remember each and every style? So instead of that, with the help of these softwares, we could be able to do the referencing as per the requirement of journals. Sometimes what happens, if the journal rejects your paper, and if you want to submit these manuscript in another journal, at that moment, simply what we need to do, after modification of our manuscript, the referencing can be done as per the requirement of that particular journal. It's so simple now. So today, the Mendeley and Zotero that we are going to see here. If you look at the Zotero, so we are knowing that this Zotero, which is a reference manager softwares, which has been created by the many academicians, and this material can be a paper, an image, or algorithm, or thesis, or anything else which can be used over here. So apart from that, so the different softwares like the Mendeley is there, Job Reference, EndNote, RefWorks. These are the different softwares which has been available, but the Zotero is quite simple as compared to the EndNote, because sometimes the EndNote, these are the paid version. The Mendeley, which provides the free software, but if you want to synchronize your data more than 2 GB, at that moment we need to pay them. So this is Zotero is an open source reference management tool. It's developed by the non-profit organization, that's a Corporation for Digital Scholarship. And this Zotero is compatible with the Windows, Mac operating system, Linux, and it is a wonderful tool which can be used and can be create references directly inside the Word. If you are using with the Windows, then the LibreOffice, or directly on the Google Docs. So this is about the Zotero. Now, what are the salient features of this Zotero? It's quite user-friendly interface. Then automatically senses web search and collects information related to the reference in a single click. What you need to do, you can organize your own collections, you can make your own library, because what happens when you get any papers, so we try to save it in our pen drive or the laptop, and sometimes we forget, but with the help of these softwares, we can organize them properly. Means for example, someone is working on the H1N1, okay, or sometimes someone is working on the nanotechnology, someone is working on the secondary metabolites, or sometimes if you're working on the e-marketing, so it has become difficult to organize them in a different folders. But with the help of these softwares, we can organize, we can save them properly by creating the different folders, okay. And so apart from that, the different citation styles, till today, more than 10,000 citation styles are available on this software, and we can simply synchronize this data. And so either you can use it offline as well as online, okay. And apart from that, when you download the various papers, you can share your library with your colleagues or the researchers, whoever needs those references. So this is what these salient features of Zotero. Now we'll see how this Zotero is being used, okay. So if you want to go for this Zotero, so this Zotero is compatible with the various operating system. As of now, we are knowing that it's compatible with the Windows, the Mac operating system, and the Linux operating system. And this can be downloaded either with the help of Firefox or the Google Chrome. Simply, you will get such type of a window when you type the Zotero, and you can download it either for the Windows operating system or the Linux operating system. And after downloading, okay, you can create a connector. That's if you are using with the Firefox, you can install the Firefox connector, or if you are using with these Chrome, you can install the Chrome connector. So with the help of these things, we can synchronize our library. And the another thing is that once you download this Zotero, you need to give a plugin, okay, plug into the Word. If you are working with the Windows operating system, so the referencing can be done with the Windows Words, Microsoft Words, And if you are working on the Linux, the LibreOffice is there. So the plugin will be downloaded. And whenever you require for referencing with the different styles, you can go for the referencing. Okay. So we'll see. So when you create this Zotero, okay, so this is what the Zotero, okay, you will see this window, right? So in this window, you may be able to see that this is having the certain icons. These are the icons once you downloaded it. So this is what the icon which is used for the creation of your new collection. Okay, so if you want to work on the the secondary metabolite, so in such a way, secondary metabolite. Okay, so I'm going to create a new folder. Okay, and if you want to find out the different references on these secondary metabolites, simply go in the Google Scholar. Okay, plants. So in such a way, you may be able to find out the different scholarly articles. Okay, for example, see here. So these are the different references regarding the secondary metabolites in the plants. So these are the PDF version which has been available. And the beauty of this Zotero software is that you can download these references, and this can be stored in those software. And apart from that, whenever you want to read those PDF version of that papers, it can be opened up, and you can read them, and you can also annotate them. Okay, if you want to give the, for example, in this hairy root culture, so this is a paper, right? So this paper, if you click on this paper, which has been already downloaded over here, so this shows that the title of the paper. Okay, what is the title of the paper, and the journal name of the journal. Okay, so it shows that the item type, which is in a journal article. The title of the paper is a hairy root culture for mass production of high value secondary metabolites, and these are the author, and this is what the abstract. So if the PDF version is not available, simply with the help of these things, you can put them into the referencing. Okay, you can use it for referencing. For example, now see here, directly I went on this the journal page. So once you click it, double click, so you can go on this journal page. Okay, and by clicking on these right click, here the different views are given, whether you want to view it online, whether you want to take a snapshot, whether you want to add a note or an attachment, or whether you want to find out whether it's available in a PDF form, right? And so many things. And apart from that, so it can also be exported. Like you must be knowing the these references can be exported mainly in the BibTeX form. Okay, so this is like the BibLaTeX is there, the BibTeX is there, right? Reference BibTeX. So these are the different referencing system through which you can download the references. Okay, for example, here if I have an article. Okay, so this is an article as I'm working on the pharmaceutical importance of this Soralia. So one of the highly medicinal plants. Okay, so with the help of this,
excuse me, sir.
Yes.
Sir, sorry for disturb, because sir, you are showing that you are online, that it is not visible to the screen, sir.
Isn't it?
Yes, sir, isn't it?
Okay. I've shared my screen, sir.
Naresh, sir, in the screen share. Sir, you are co-host, so you can share this.
So I have already shared.
But only the slide of PowerPoint presentation is there. The online that you are showing, that is not visible.
Is it? Okay. Okay. So the entire screen. Okay. Yes. I hope now it is visible. Yes.
Yeah.
Sorry, I've forgotten that. Okay. So we are talking about this Zotero, right? Now I hope it's visible now. So once you go for this downloading this Zotero software, okay, you will get such a screen. Okay. So with the help of this Zotero software, you can create your own library. Like this, you can see at the left side of this screen, my library is there. If you click on this below this file, this icon is used for the new collection, whether you want to create your new collection or a new folder. Okay, so I have already created 2 folders, like the hairy root culture or secondary metabolites. And apart from that, once you download these papers, so there are the chances of duplication of different references. Okay, so you can with the help of these duplicate items, you can find out whether the references are duplicated or not. Okay, so if you go on this for hairy root culture, this is what the reference or the topic on which I would like to work on. So this is the paper. Okay, and if I click on if I double click on this paper, it will bring you on the website of that particular journal. Okay, so here you can go, you can see the abstract and the other things. And apart from that, when you find out how to download the citations, so if you like to download the citations instead of going with this tedious task, you can simply go into your software, like this Zotero, and with the help of Google search, you can download the various paper. For example, here the influence of abiotic stresses. Okay, so this is what the paper, and once you download it, okay, so look at here. So this is what the paper, and if you go with the Zotero connector, this is what the Zotero connector, it will add into your Zotero library. Okay, see here the influence of the abiotic stress, it is appeared now. So I was stored in this heavy. So in future, if you want to read this paper, you can simply click on it. Yes, in such a way you can go it. Okay, and the PDF version is available, and this PDF can be stored, yes, to Zotero connector. Likewise, yes. Yeah, hairy root culture. Click on it. Okay, done. It is done. Okay, so in such a way it can be done. If you further, we can go for the referencing. How we can go for the referencing? See here, this is a paper I would like to work on, right? So there's the pharmaceutical activities and effect of various abiotic stresses. Okay, elicitors on bioactive constituents for Sorelia corallifolia. It's a Bakuchi. So for example, so if I want to put my reference over here, okay, this is what the reference is. I'm going to delete it. Okay. So you'll find out if you open the Microsoft Windows, this Zotero plugin will be seen here. Look at here, this Zotero plugin. Okay, so with these Zotero plugins, you can go for the referencing, right? So what type of reference do you want to see here? Yes, sir, is it visible now?
Yes, sir, it is visible.
Okay, so let me tell you one thing. Once you download this Zotero, you have to download the Chrome extension or the Firefox extension, and then subsequently you need to download the plugins for the Microsoft Word if you are using with the Windows, or the LibreOffice if you are using with the Linux. So when this plugin or this icon will be appear in your Microsoft Word, click on this Zotero, and here these different icons are there. It's a add citation. If you want to put the notes on this paper, like the add note, okay. So okay, so these are the different. So what type of paper has been there, or the different style is there. So add citation. Okay. So in add citation, look at here. There's so many different types of styles of referencing is there. So mostly the elsewhere type. Okay, the Harvard, the IEEE has been there, the Nature, and so on. And if you find that the style which your journal needs, it is not available here, so you can simply put on these manage styles. Okay, if you put on the manage styles, you will be able to see them. Okay, different. Okay, so the style editors are there. Okay, so different style editors. With the help of these style editors, you may be able to find out the different types of styles. Okay, so in that case, so I would like to go for the IEEE format. Okay, this is the IEEE format. Okay, and I want to give the reference. Okay, from this one. I would like to put my reference over here. So what I will do here, I will click on the add citation. So this window will appear here for the Zotero, and you will find here these so these are influence of abiotic stresses, and this is what the paper to which you want to give a reference. So I will click over here, enter, yes. Okay, again, if you want to give the second reference over here somewhere, okay, and then again add citation. So add citation, and again this Zotero link will appear over here. Okay, and you just put here something that this Sorella corylifera Kustanshi, okay, which has been already downloaded. Okay, and this you want to cite over here. Put an enter. Yes, this is what been there. And once you're done with all the references, okay, whatever you want to put in your paper, you then come to the add to bibliography. Okay. See at the bottom. So this type of so in this way it will come down. So these are the different references that are going over here. Okay. So these are the references. In such a way you can go with your things. Okay. And another thing is that, so this is a very, and see this Zotero is very useful, and by practicing you will be expert in this Zotero. And my aim for this webinar is to just have a glance and how these reference softwares are used. Okay. The another one is that here the next one, the reference tool that we are going to see here, the Mendeley. Okay. So this is another type of online referencing tool. See here in the Mendeley is again the can be worked online as well as offline. And why we need to go for this Mendeley, which it also organizes your documents. It also makes the groups for working with your colleagues. You can also access papers of a similar them as yours. Means what? If you want to share your paper with your colleagues, or your colleague is having certain papers which you want to access for your future project, it can be shared with each other. And this Mendeley citation, it's also used for the Mendeley citation, because the Mendeley is been developed by the Elsevier publishers, and the different journals which has been hosted by the Elsevier publishers, they usually recommend for this Mendeley. Simply what you need to do here for this Mendeley as well as Zotero, you have to log in after downloading these softwares with your authorized email ID, and you can go for this citation. Okay, and repetition of papers can be removed, as many of time what happens, we put the references repeatedly, and sometimes we get fed up with this referencing. At that moment, the software which helps us and which shows that this reference is repeated, you need to remove it. Okay, and apart from that, it supports the cloud system. So this library can be accessed from anywhere with the help of your cloud storage. And so this software is also having the two type of memory system. One, the desktop version, which is having a short-term memory, means whatever is available stored in the desktop, that is stored there. But if you connect with the internet, okay, and you sync your library, so that can be accessed from anywhere in the world. Okay, so this is what the beauty of Mendeley. So for downloading this Mendeley, the window will appear like this: Mendeley Reference Manager for desktop. Okay, it is also supports for the Mac, Windows, and Linux. Okay, and once you download it, okay, once you download it, so simply again you will have to give the plugin. Okay, for this Mendeley software. Okay, this is a Mendeley. See this. This is the screen. It views like this. So I already logged in with my email ID, and I have downloaded certain references. Okay, and again, if you want to download some papers, okay, like this. Okay, this is a paper you want to plug in. This difficulty for this. Okay. Okay. So in such a way, you can yes. Okay. So in this Mendeley also, you can go with this Mendeley. Here there are the file. Okay, you can here with the help of this file, you can also export the files like the bibtex or endnote. If you're already having these references, with the help of this bibtex or the endnote XML or the other referencing system, you can access them in this Mendeley, or you can add new. Okay, simply click on it here. You can create a folder. Okay, or you can access it directly from the computer which has already been there. Okay, and these are the certain papers which has been downloaded it in this Mendeley. Okay, and if you double click on it, simply it provides you, look, this is an entire paper like the traditional uses of Southern African medicinal plants. Okay, and in the right side, it gives a brief. That's the metadata shows so what type of paper it is. See here, it's a journal article. Then it also shows that the year of publication, okay, the author name, and the what citation style has been used here. So either you can go with this paper, okay, simply, And you can go back to the end note. Yes. Yeah, you can come back from here. So I have created the two folders for the ethnobotany and the for the Soralia. If I want to see only the papers which are related with the ethnobotany, yes, once you click it here, so the ethnobotany related papers. See, these are the certain papers which I have downloaded for the ethnobotany work, and for the Soralia. So these are the papers which are exclusively downloaded for this Soralia. So again here, if this can be shared, and once you download it, okay, so don't forget to synchronize it. Okay, so this is what the sync button, you need to synchronize it. So that means whatever the papers you have downloaded, it will be uploaded on the cloud. So up to 2 GB Mendeley provides the space, so that much of data is quite sufficient for saving your papers, and it can be accessed from anywhere. Okay, and apart from that, so if you do want to go with this paper, suppose I want to work on the orchids. Okay, so this is a paper. Okay, I want to work on it. Okay, so just after writing here is the references. Okay, I hope so that you might be understanding. Okay, simply here again, after downloading this Mendeley, once you downloaded the paper from the Google's or the any searching tools, here again you will see the reference icon. Okay, once you go in the reference icon, there is a the Mendeley plugins will come up here. Okay, it will pop up. Okay, once you click on the reference, so this is what the Mendeley citation. You go here and want to put the reference over here. For example, if you want to give here reference, that put in a reference, click on here, and here are the different styles. So what styles would you like to use? Okay, so whether it's a Chicago or the American Psychological Association, IEEE, or the Nature, okay, or Harvard. Okay, so this is the APA system. So with this, you will be able to find out the different references. See here, the Mendeley site is opened up. Okay, and here are the citation styles. What type of citations would you like to use? So different citation styles are there. So if you want another citation style, you can simply put any journal name like the PLOS one, okay, or the PLOS biology. See, I'm going to communicate my paper with the PLOS one. Let's put it here. It will update. Yes, add citation style. Okay, now close and reference over here. Okay, so put it here. Okay, but just where you want to give the reference, you just type here this search here. In that case, if you want to search so this one, if you want to give over here, okay, just put it on here, give the citation. Yeah, it has been here. Okay, and once you put it here, so again you can click on here and you want to give the reference somewhere here. Okay, and is so this is what the reference you want to give her, the traditional uses of this plant. put the citation, insert the citation. Once we insert it, and then come back to these references down, and once you've done it, so go in the more, insert bibliography. Okay, so continue. Yes, so this is what the reference. You need not to worry about these their DOI number, their journal title, then the page number, the volume number. You forget about it. Okay, everything is done with the help of these online software, these reference tools. Okay, so this is how you can go with this reference management system. And sometimes many a time we our paper gets rejects, and the most tedious job is the referencing. So this software will help us to make our references according to the journal style. So there are so many different journals available on the different publishers. Okay, and the styles, more than 10,000 different styles are available. So any style the journal follows, we can follow them, and accordingly we can do our referencing. I hope so that you could understand the basics of this online reference software, and you can practice it by downloading these softwares, and you can also share these softwares with your friends. Okay, so this is what, that's all. Thank you very much.
Thank you, sir, for such a wonderful and fruitful knowledge using different softwares like Mentoring and the Zotero for referencing. And most of the things that I also don't know, I got the idea for referencing. Now the session is for question and answer. So participants are requested to ask the question to resource person or give your questions in the chat box. So we will directly ask to the resource person and he will sort out your
So you can put my email ID, you can share with them if they want any kind of help or suggestion. So I will be very happy to help them out.
Yes, sir. Yes, sir. I will.
Because
Hello.
within, yes.
Hello, Abra sir.
Yes, sir.
Okay, thanks for this very informative and very important talk. I really appreciate that you chose to give this reference management talk here. So my question is, I have some questions. My question is, what are the other softwares you can suggest for the students, that can be useful for their research in future? Can you please suggest a few more?
Yeah, see, apart from this Zotero and the Mendeley, the EndNote is there, which has been widely used by the researchers throughout the globe. That one has been there, but the problem with the EndNote is that it doesn't provide you the sufficient space for storing your references, and it is they are having the paid version. But in case of this Mendeley and Zotero, these are the free one, and very easy to use. But those researchers or the students who are pursuing their PhD, I will suggest them to go with the LaTeX. One more software is there. LaTeX is another software which is freely available. You just have to visit the Overleaf website, okay, www.overleaf, and you go and register yourself on this Overleaf. You can directly you can type your thesis over there. You can type your paper. You can prepare the presentation, or you can make your poster presentation. It's a wonderful software. I will suggest, but again, the thing is that you need some time to understand. You need to practice it properly. So then you will be very
Okay,
easy
and
to handle with it.
anything would you like to add about some other plugins like Grammarly, for example?
So Grammarly is a wonderful thing. So those sometimes what happen, our Indians problem is that we are actually we are very perfect in grammar, but nowadays by continuous use of this social networking, and we are using the shortcut keys, right? And we have forgotten about the grammar perfection. So Grammarly will help you to correct the sentences which make it corrected with the grammar. Okay, grammatically perfect one.
Thank you.
Thank you very much.
Thank you, sir. Sir, one thing I want to mention here that
Yes.
He is our another resource person, Dr. Azarian Shakespeare.
Yeah.
And one more thing that we all three are connected with the Government Institute of Science Aurangabad.
Oh, so nice.
Because Azhar sir is also the alumni of our Government Institute of Science.
Okay.
You are also there, and myself also there.
Azhar sir was in Botany department or some other department?
No, sir. He is from Geology department.
Okay. Now the second resource person.
Second resource person. Yeah.
Yes. It was
And,
nice.
so this is a meeting for the alumni of the Government Institute of Science also.
Absolutely. It's nice.
Nice
Thank you
to
very much, J.A.T too.
Nice to meet you.
Same here.
Yeah. So thank you, sir, for such wonderful presentation, and some you had given hands-on training also. Which way that we will use the referencing tools for writing our manuscripts publications. Thank you, sir. I am very much thankful on behalf of our J.A.T Trust, J.A.T Senior College, our principal, our academic administrative coordinator, IQAC coordinator, from all of and participant also. So I am very much thankful on behalf of all of these to you, sir. Sir. Thank you, sir. Thank you very much.
Thank you very much, sir.
One more thing I have to mention, sir. I hope that you will be offline in our college in one day, because from last 1 year we were trying to meet you, but there are lots of conditions that are not making success.
Due to pandemic situation, we could not make it possible to come over here. So in nearby future, inshallah I will come over here.
Yeah, sir. Now you are very near to us, because Gadchiroli was so far, sir.
So far, absolutely. Thank you very much for giving me this opportunity. So this was a wonderful platform, so that whatever the techniques which I have acquainted during my due course, so I could make it possible to share with the other people, so that they can take the benefit of these softwares.
Thank you, sir. Thank you very much.
Thank you very much. Thank you, Azhar sir.
Thank you.
Now we will move towards our next session. For the next second session, we have a resource person, Dr. Azarian Shakespeare. So now I would like to call Professor Hadia from Department of Botany to introduce our next resource person.
Dr. Azarian Shakespeare is a postdoctoral fellow in Department of Geology, University of Johannesburg, South Africa. He had done his PhD under the title Mineralogy of Selected Kimberlite Sand Rock from the Vajrakaror Kimberlite Field, Southern India. He had done his diploma certificate course in hydrology in 2009 from Aurangabad, India. He had done his BSc in 2010 and MSc in geology, and he was gold medalist and university topper of He had done his PhD in 2010 He had done his BSc in 2010 and MSc in Geology, and he was gold medalist and university topper of Dr. BAM University, Aurangabad, India. He experienced as summer trainee at Department of Geology and Geophysics, Swadia Institute of Himalayan Geology, Dehradun, India in 2011. He also experienced as a junior research fellow at Department of Earth Science, Indian Institute of Technology, Mumbai, India. He also experienced Grant India in 2015. Also received the Best Paper Award in Mineral Characterization category at MPT 2016. He granted as International Kimberlite Conference Financial Support 11 IKC in 2017 and also granted as International Travel Support Scheme SERB DST in India to 2017. He received 35th IGC Foundation Legacy Fund in 2019 and he published his research papers and articles in 7 different publications at Tanzania, London, India, etc. He also presented 2 posters in different conferences and also 3 oral presentation and submitted the abstract at International Geological Congress 2020 in Delhi, India.
Thank you, Hadia ma'am. So now I would like Azhar sir. Session is handed over to Azhar sir. Sir, start your presentation.
Okay. Okay, thank you. Thank you for this nice introduction. Actually, I wanted to introduce myself, by myself, but it's okay that, I got such a good and nice introduction here. So can you see my presentation?
Yes, sir. You are visible.
Okay, just hold on. Okay. So, hello everyone. Juma Mubarak, and thank you, Dr. Yahya Khan, for inviting me for this nice seminar series. And I also thank you all of the dignitaries on the dais and all the students. And here I will talk about some spectroscopic techniques in geosciences, which are also related to biosciences. And I hope that you will find some applications about about the subjects of your own interest. Okay, so as you can see that my topic is spectroscopic methods in geosciences. I will give you some introduction and we will go through some at least 2 to 3 different spectroscopic techniques. So I am Dr. Azhar Mohammad Sheikh. I'm a research assistant at University of Witwatersrand. Earlier, I was postdoc at University of Johannesburg in South Africa, and I finished my PhD from IIT Bombay. This nice picture in the background here is nothing but a diamond mine, which has been abundant in the last 10 years back in South Africa. So we visited this diamond mine for collection of samples. So there are 2 kind of geologists, I would like to say. One geologist, those who go for field work, those who go for collecting the samples in the field. And the other geoscientists are those work mostly in the lab. So those are mostly the lab scientists who work with the instruments. So those are instrument scientists. So I'm more kind of instrument scientist. That's why I chose to give I chose to share my knowledge about instruments to you. I hope that you will like this talk, and yeah, please keep if you have any questions, please ask me. You can ask me in the middle, you can ask me at the end of the presentation as well. Okay, so let's move on to first slide. So about myself, I told you that I wanted to give some introduction about myself, but it's fine. So I did my master's in geology from Dr. Babasaheb Ambedkar Marathwada University. As Dr. Yahya Khan said that we are the alumni from the same institute, Government Institute of Science. So I did started my research career as a research fellow at IIT Bombay. I was involved in establishing a instrument which is electron microprobe analyzer in IIT Bombay. So I got trained there for this instrument, and then at that moment of my life, I decided to become a life scientist instead of field geologist. So I did my PhD in 2018 from IIT Bombay. I worked on diamond-bearing rocks from southern India. So you will know. And then I started my post-PhD research career from University of Johannesburg when I joined in 2019 in University of Johannesburg. And then recently in January, I started working as assistant researcher at University of Witwatersrand in Johannesburg. So these are my publications. As you can see that my publications are, are mostly from India and as well as Africa. So I work in southern India on diamond bearing rocks that gives us the diamond. Those rocks we call them as kimberlites, and kimberlites are located in southern India, mostly from Andhra Pradesh. And as you can see that my research has recently been shifted towards the South Africa. I worked on Tanzania, South Africa, as well as Congo. So I also attended few international conferences. For example, this one here, the Goldsmith, very prestigious conference for geoscientists, which was held in Prague, Czech Republic. Then I attended International Kimberlite Conference and Diamond Conference in Botswana. Then I attended another Goldsmith conference in Barcelona. And then last year we did, due to the COVID lockdown, we did attended the virtual conference from that was, held from France. And then this year I'm going to attend the conference again in Hawaii. So I also did some workshops like doing some melt modeling, thermodynamic modeling, and instrument related workshops. So lots of things about me. Let's move on to the subject. So chronology of this talk will be, I will give you some introduction about the geology and geosciences, and then I will move on to the different spectroscopic techniques which I'm going to discuss here. So as you can see this figure here, very nice sedimentary rocks has been deposited. This is the image from Grand Canyon, USA. So the geologists, if you are geologist, you will always, get hooked about these kind of features in the field when you are go for the field work. So what is geosciences and what is geology? So there are two things actually, the geosciences and geology. The geosciences are actually, it's a broad field. It also includes the geology plus meteorology and oceanography, whatever the research about the Earth. So Earth, for geosciences, it includes also the climate and also the water on this on the ocean, and whatever the biomass, if there is some link with the Earth or related to the rocks. And the other subject which is I'm more related is the geology, which is mostly related to the scientific study of Earth and forces acting upon it. And for this, I showed here an illustrative image. And I hope that my friend Yahya Khan will recognize this image because he has very, interest in images and the videos, and he's very interested in Turkey. And this image is from Turkey, which is known as the Cappadocia. It's a sedimentary feature. It's an erosional feature actually. So there is this limestone rocks here, and which has been get eroded, and you will find this kind of columnar structures in Cappadocia, which is very world famous and global heritage. So the geology is related to these rocks. What are these rocks? What is their composition? How they form? When they form? Their ages? Their depositional ages? What kind of fossils we can find there? And also the forces. What kind of forces are acting to make these features? For example, the erosional features. So whether there was activity of the water, whether there was activity of wind. So these kind of courses, this all combined study gives you the geology. I hope that I'm not going too fast. If you want me to go a little bit slow, you can always ask me. Okay. So I hope that you are getting what I'm talking. Okay. So for geology, in geology, there are different subjects like igneous petrology, sedimentary petrology, metamorphic petrology, fossil science, and lot of things. So in that, I'm related to mostly I'm related to geochemistry, and geochemistry is nothing but another branch of chemistry or geoscience. It's interdisciplinary science between the geoscience and the chemistry. So the chemistry is mostly about the composition and constitution of substances that may undergo any changes. So it's all about the composition elements and isotopes, all those things. But for geochemistry, we just talk about mostly, about the planets and Earth, including Earth and their composition. Okay, and we also talk about their ages, if there is some ages or if there is some isotopic changes. Okay, so for example, here, if you see on the right side panel here, this diagram, which is the cross section of the Earth, that shows Earth has different layers of composition. For example, there is core, there is mantle, there is crust, which are based on which are classified based on their chemical composition. As you can see here, the core is mostly rich in iron and nickel, mantle is mostly silicates, and the crust is mostly granites and basalt. Okay, so the depending we study the internal composition of the Earth as well as whatever the rocks we find on the surface. And on the left side panel here, you can see that there are nice layerings in sedimentary rock, which is sandstone. So all these layers, thin layers, that can give us information about the environment, what kind of environment was there when these kind of sediments were deposited. So we study these layers as well as their composition. Okay. So moving to next, the geochemistry. In geochemistry we do geochemistry. If I want to know the composition of particular rock, for example. Okay. So we try to know the information. We try to get the information of that particular rock composition by using either bulk rock. Bulk rock means you take a rock as a whole and you crush it without considering any kind of heterogeneity, any kind of layering or whatever is there. Okay, so you crush the rock, make the solution, and analyze that rock by using either Icpms or X-ray or XRF. Okay, so that composition will give you that rock composition will be as a whole. What is the composition of that rock? For example, this kimberlite here, which I already told you, a diamond bearing rock. This is from southern India, Vajrakarur. You can see that this diamond bearing kimberlite has very fine grained matrix, and it also has some oval shaped, circular oval shaped minerals, which is nothing but olivine. And these minerals, if you see, if I crush this whole rock, I will know the composition of this rock as a whole. Okay. But what if I just wanted to know the composition of these small grains, just the olivine and not the matrix? So what I can do? So for that, either I can separate these grains like this at the bottom here. These are some garnets in very nice color from violet to orange and red. And there is this planoparoxene, which is in green color. So you can see that in garnet and planoparoxene, there is much difference in their colors. So these colors, nothing but representative of their composition, because composition changes, the color of the grain changes. Okay. So now, if I want to know the individual grain composition, then either I can crush this rock and then separate out those grains from this kimberlite gold rock, and then analyze them and then I will know that what is the composition of garnet and what is the composition of planoparoxene. But there is another method that I can use. It is simple, and it doesn't need to. We don't have to crush the rock. We don't have to separate those grains. That is called as in situ mineral analysis. So we make the thin sections out of this rock, and we can just with the help of microscope, we can point out our area of analysis and take the analytical data for the particular area. So for example, as you guys are from biosciences, you need some bioscience scientific examples. So for here, I have this image, color image for this brain, human brain, and there is color image of this watermelon. So as you can see that if I want to know the composition of this watermelon, so there are two things. Either I can crush this watermelon as a whole and get the composition that will also include the composition of this red material as well as the grain as well as the crust of this watermelon. Okay, so this will give me mixed composition of the watermelon. Similar for the brain, if you want to know the composition of the brain, you can either take the brain as a whole and analyze. Okay, so you will get the mixed composition of the brain, but you will never know the you will not know the composition of this particular area of the brain or component of the watermelon. If you want to know the composition of just for say the seed or just for this say this crust, then you either you need to separate it out or you can make the thin section and you can analyze under the probe. Okay, so that's the in situ mineral analysis. I'm going to talk about this method in next few slides. So for spectroscopy, as you guys may have known, the spectroscopic methods are of two kinds. One is wave dispersive spectroscopic techniques, other one is energy dispersive. So what is the spectroscopic techniques? Is actually what we're doing there. So what we are doing is we are generating a spectra, a spectrum of that contains a lot of information about the composition, about the physical characteristics of that particular matter, about the crystal structure, about the whatever things that beam kind of whatever the energy is interacting with this matter. Okay, so that particular spectrum contains a lot of information. We need to separate it out. Okay, so just like the prism, when we put the light, so the normal light on the prism, the light separates out, disperses all the colors, and then we know that different colors are coming from one single spectrum of light. Okay, so here when we get the information from either Icpms, either from XRF or EPMA, we need to separate out the spectrum and analyze each and every spectrum for getting the different information. For example, here, as you can see the spectra here, we use X-rays for getting the information about the composition of particular. So the WDS, there are some differences in WDS and EDS. WDS is dispersion is based on the wavelength of particular spectra. EDS is based on energy. And then in WDS, we use the different crystals, the synthetic crystal gratings to make that spectra disperse. And for this, we use Brack's law. And because when you if you disperse the light on a grating, you wanted to know which wavelength will fall on which area or which particular area, what is the angle of that dispersion, so that you can measure the secondary X-rays. And then there are some advantages. The WDS is have more resolution because this wave dispersive is very convenient and very it's very effective technique to make the dispersion, unlike the EDS. So for EDS, we use the dispersion based on the energy of this particular atom or whatever the say photon. So that's the energy dispersive. We use charge per mass ratio sometimes, and we also use this will give you relatively low resolution because it doesn't separate out the spectrum in the horizontal way. It separates out the spectrum based on the speed. For example, how much which particular atom or which particular element is moving fast in that particular field. Okay, and this one is relatively fast because you can just scan a lot of elements in a matter of seconds. Okay, so moving to our techniques, I will talk about first the X-ray fluorescence spectroscopy. As you can see, these two images here, there are two different kind of XRF available in the market. One is the this big one which you can install in the lab, and the other one is portable. So the big one, as it has much more advantages because you can analyze more and more elements with higher resolution and higher accuracy. While this portable one will give you low accuracy and low resolution. So but it's also useful because you can take it to the field and you can analyze whatever the preliminary composition of that particular rock at the field. You don't have to bring the sample in the lab. So for XRF, what we do is we generate the secondary X-ray using a primary X-ray source, and the secondary X-ray are actually characteristics of particular element. So when we bombard the X-ray on a particular matter, it will generate the secondary X-ray, and that secondary X-ray will give us the composition of particular matter, what kind of element is there available there. So this technique is very fast. It gives you analysis in matter of seconds. It is non-destructive because if you make one sample, that sample you can analyze for 1,000 times. It doesn't destruct your sample. It's always you can always reanalyze that. Okay. It requires minimal sample preparation. You don't have to go for very tedious and very hard kind of difficult kind of sample preparation. You just cast the rock, and you can make the either solid beads out of it, or you can also make fusion glass. Okay, so those are very small easy techniques. You can also analyze solid, liquid, and as well as gases, but as per your requirement. Okay, so just hold on. I just moved to some different slide. Sorry. Okay, sorry for the interruption. Okay, so if you talk about the theory behind the XRF, if you see here, this is the atom. For example, this atom was inside the sample of which one you are going to analyze here. So we know the atom consists of nucleus. It consists of different orbitals that has different electrons. So for example, this atom here, when you bombard the X-ray, that's the incoming X-ray from this side. And when this X-ray bombarded on the atom, it dislodges the electrons from the lower energy shell. For example, this one here is this K shell, which has lowermost energy, and then depending on the how much energy of the X-ray you are bombarding, the electron will get dislodged. That's the free electrons now. And then that there is tendency of every atom, when you discharge the electron from a lower energy level, the electrons from a higher energy level, they jump onto the lower energy level to satisfy that lower energy level shell. And then that's what happening here. As you can see that there is electron get dislodged from the K shell, and then there is electron is coming from L and M shell. Okay, so if you see if there is jump of electron from K shell, then you will have the K alpha line. That's the secondary, because the higher energy level electrons will have higher energy level, and when it is jumping from higher to lower energy level, it has to emit some energy. It has to do some energy, and then it can come back to this lower energy level. So when it's jumping from lower energy level, it will give you K line. And if it is coming from M energy, M shell, then it is giving K beta line. So every interaction, every atom has different lines of these X-rays. So these are secondary X-rays. This one is primary. So you can analyze either K alpha line. You can also analyze K beta line. As you can see here, there are a whole bunch of X-ray emissions. You can see that there is K alpha line, K beta line, L alpha line, M alpha line, and there are whole different lines. Okay, so as you can see that there is if there is a jump of electron from from L shell to M shell, there is highest energy get released. So you will have K alpha line, which is very high which will be of higher energy. So it's easy to detect the K alpha line than compared to, for example, say M alpha line, because there is very small energy difference between M shell and N shell. So when there is jump of electron from M shell to N shell, then there is very small difference. So it's very difficult to identify that line. So usually we use the K alpha, K beta line, and depending on if there is some overlap, we need, if we don't have any options, so we choose the different lines of higher energy level as well. So the XRF has different parts. For example, there is XRF chamber. There should be, XRF chamber should have some particular kind of vacuum, because you don't want any kind of element or any kind of gaseous material to come into that path of the electron, so that will, charge the of that molecule or atom, and that can give you different composition, and then you will have a lot of background effect. Okay, so there is chamber which is maintained under the vacuum, and then there is this X-ray tube, that's the source for X-ray that emits the X-ray, and then there is sample holder. You need something to hold the sample. Then there is this colorimeter. I will come on to the colorimeter later on. That's the colorimeter here, which measures the your spectrum. And then there is crystal grating. As I told you that you need crystal grating to disperse the spectrum of X-ray, so that you will get some, you will get different X-rays from different elements. So there is detector here. You measure the each element in the detector and computer system. Okay. So the working is like this. You have this detector, you place your sample over here, that's on the top, and you put some crystal gratings. So when you shoot out your X-rays, it will bombard. That's the other way actually. That's the X-ray tube here. When you shoot out your X-rays on the sample, that will bombard on the sample, that will generate secondary X-ray. That X-ray moves towards the crystal grating. And crystal grating, what it does is it separates out your each X-rays from different elements, because these secondary X-rays are characteristics of each element. For example, there is alpha line for sodium, alpha line for barium, alpha line for silicon, magnesium, nickel. Each element has different wavelength. Okay, so each element has different energy and different wavelength of X-rays. Okay, so based on the crystal grating, we separate out our spectrum, and now we know that particular wavelength of element, wavelength of the spectra represents some particular element. So based on that, we move our detector like this in circular manner, and we detect all the elements one by one. So that's the Rowland circle here. As you can see, that electron is coming from the beam. That's bombarded on the sample, that generates secondary X-ray. That's the crystal grating, and the crystal grating is usually a circular shape, so that that gives direction of the secondary X-ray towards the detector. Okay, so this crystal grating moves like this in the circular manner, so that different elements can get covered. So this spectrum, as you can see, has a broad spectra here. And then the crystal sends the secondary electrons towards the detector. Okay, so detector measures the electron here. So both crystal and detector moves in circular manner. Okay, so what is the theory behind it? Okay, so how you know that particular, for example, sodium alpha line, where it will fall? Because when you are putting the grating here, for example, go back. If I wanted to know that, I want to analyze the sodium from the sample, it will generate the full spectrum. The spectrum, we don't know what kind of elements are there, because there is also there's sodium, nickel, silicon, magnesium, calcium, whole bunch of spectrum is there. Now we want to separate the sodium. How you will do that? So we put the crystal grating here, and then we know that for sodium, the X-ray, secondary X-ray line, for example, K alpha line has particular wavelength. Okay, so that's the lambda here. So we use the Brack's law. I hope that you are aware of this law. So the law states that if there is a spectrum, for example, the sodium alpha line here, that's this lambda is equal to 2d sine theta. That's the twice of d sine theta. D is the space, internal spacing between the atoms of the grating. So what we do is we make this grating synthetically. There is a reason, because we want to know what is the exact distance of the interspatial distance between the atoms. So for this grating, we know this d distance here, and if we know the d here, then we would like to know the theta. For example, for K alpha line of sodium, I know the lambda for K alpha line of sodium. Then I will put the K alpha line wavelength of the sodium here. Then I know the D, the spacing between the the grating, and then I can calculate the theta. Theta is nothing but when it is coming, the X-ray line is falling here, and then it is diffracted on the surface, and then we would like to know what is this theta angle where this the sodium K alpha line will fall. So that's the theta angle that we can calculate using this equation of Brack's law. Okay, so we calculate like this. That's as simple as like that. So your detect the sample is putting X-ray emitting the secondary X-rays falling on the grating, and then there is this theta. For sodium, for example, theta is 30 degree. For magnesium is 20 degree. For silicon is 10 degree. Okay, so there is whole bunch of elements will separate out on this line here. Okay, so you can move your detector. For example, for magnesium you move to the left, for example right, and for calcium, for example, you can move to the right. Okay, so depending on the theta angle. So when you do this, you need something, how would you know the intensity of particular line? For example, K alpha line of the sodium intensity is proportional to the concentration of sodium in that matter. Okay, so how would you know that what is the concentration of that particular matter? So we have to have some standards. That's the procedure for calibration, right? So we have to have some material that will have the composition of known composition. For example, if I measure the some material which has, for say 10 ppm of sodium, okay, and then you can calibrate using this what is the intensity of for 10 ppm in that matter. Okay, so if you get the intensity of particular unknown, then you can relate with that and get the composition of unknown. So that's the equation for this. The Ci, that's the concentration of particular element in element I in matter 1, and divided by concentration of matter 2 of element I. So that's equal to the ratio of intensity of matter of element in matter 1 and matter 2. Okay, so that's the relation. So but it's not as simple as as you see in this equation. There are lots of things coming in because there is also background effect, there is also matrix effect that has to be cancelled out. Okay, so that's all those things will be taken care of by the software. Okay, I'm not going to go into that detail. Okay, so there is another technique which is Icpms. That's the inductive coupled plasma plus mass spectrometer. So this instrument has two different parts. One is the inductively coupled plasma and the mass spectrometer. So this instrument is fabulous. It can measure the element from ppb level, parts per billion level, to weight percent. Okay, so if you want to analyze some elements, like for example any element which has very low concentration, for example iridium. So iridium is very rare element on Earth. It only occurs in the meteorites. And for example, if you want to analyze some isotopes, for example, say rubidium 87, strontium 87, neodymium 144, 142, or uranium isotopes, lead isotopes, any kind of isotopes you can measure by using the Icpms. Okay, so here what is happening actually, so we are this is the technique is destructive. Destructive means you can, if you analyze a particular amount of sample that get destructed, you cannot analyze the same material, but you need some other material to analyze. Okay, this is very rapid multi-element analysis, very low detection limits. You can go up to the parts per billion here. It has very low inter-element interference, and there is a whole bunch of new techniques coming in if there is some interference that you can use. Okay, there is wide range of elements that you can analyze, from elements to isotopes. As I already told you, there is higher degree of atomization, that is the sample generates atoms, and that gives you stronger signal. Okay, so how does it work? So as I told you that the instrument consists of two things. One is coupled plasma, okay, Icpms. So what is the inductive coupled plasma? The inductive coupled plasma is nothing but a plasma, which is fourth state of matter, as we all of know. So what we do is we use the argon gas, and we put this argon gas in this coil here in the field, which is the RF coil, which is nothing but the resonance magnetic field, which gives which generates very inductively generated electromagnetic field, and that generates the converts the argon into plasma, because in this field the argon gas, they couple each other, they bombard on each other, and that will form generate the plasma. Okay, so this plasma has temperature of around 6,700 degree Kelvin. Okay, so that's very high temperature. If you put anything into that, that will get atomatized. So you will form whatever the matter is there, it will disintegrate into the ions and cations. Okay, so what we do is we introduce our samples into this plasma that dissociate that matter in the form of atoms, and we take these atoms into the mass spectrometer, and that mass spectrometer measures this atom. So here this spectroscopy techniques is completely different from what we were doing in XRF. That's WDS. This is the kind of EDS technique, which measures the atoms directly instead of their secondary X-rays. Okay, so what does it have? So it has the sample technique here is a little bit complicated, because you need to create a homogeneous solution, because as I told you that rock or particular matter, for example, watermelon can have different zones, can have different smaller parts inside it. So you cannot analyze for example particular one. So if you want to measure the whole rock, then you need to crush them, homogenize them, and then the best way is to crush those samples and make it in solution, because solution is always homogeneous, and introduce those samples in the form of solution into this plasma that will generate the atoms, and that you can transfer this atom for analysis. Okay, so there are a whole different bunch of sample analytical techniques. For biosciences, there may be some different one. I'm not aware of. Okay, so for ICPMS, there are different parts. Obviously, argon plasma. This one is under very high vacuum. As I told you, every instrument has vacuum. And then there is this plasma chamber. We use the argon. Somebody can ask questions that, why we are using argon. So because the argon we are using, because it's inert gas, usually it doesn't interact with any atom. It doesn't give you any compounds that we don't want to analyze. It doesn't consume your material, because if it is analyzing with reacting with sodium or some kind of atoms, then what will happen? You are losing some matter, okay, in this reaction. So argon is inert gas, and it's also it doesn't gives you interference. You are not when you are doing this analysis, mostly it's likely that you are not going to analyze the argon. So it's very convenient to use the argon gas. Okay, so there are some other gases which are also used. For example, we use helium, we use what is that, the other one, nitrogen gas, to increase the sensitivity and the more say analysis, the accuracy of the sample. Okay, so there is plasma chamber, argon plasma chamber. That's the torch here. You can see that that's the ICPMS torch that generates the plasma from argon. And then there is this sample introduction system. That's the sample introduction system that sample. There are some few complications. How you want to put the samples inside, because you cannot dump anything and in any kind of style. You need to have some aerosol of the liquid that will give optimum and maximum, I would say maximum atomization for analysis and better atomization. So for that we have this sample introduction system. We use the spray chamber, nebulizers for introducing the sample. We use argon as a carrier gas that carries this atom, because if you're introducing, you need some gas to carry this liquid and aerosol into the plasma torch as well as in the mass spectrometer. Okay, so and then there is this mass spectrograph. That's the mass spectrograph here that that acts on the different electromagnetic field or magnetic field that will separate out different elements based on their charge by mass ratio. Okay, so there are some detectors and computer system. Okay, so what is the working of this Icpms? Okay, so when you can, if you see this diagram here, you have this solution which you crush your sample and made a homogeneous solution, and then you introduce your solution into the spray chamber, and then the spray chamber get, there is argon gas coming from this side, and then this sample get introduced into the plasma here in greenish blue color, and then this plasma will dissociate all this matter into the ions and cations. That cations will be moved towards the spectrometer by using argon gas flow, and then this here, what will have there will be few lenses to remove some undissociated material, or whatever the interference material that we don't want to analyze, that will be separate out here. Then there is this electromagnetic field that will change the direction of that particular atom or isotope, for example, because every isotope, for example, oxygen 16, oxygen 14, oxygen 18. So oxygen isotope has each isotope has different mass. So each isotope has different mass because it has additional neutrons in the nucleus that gives you additional mass based on the charge, because the charge is same for but there is a difference in their mass. So based on the charge per mass ratio, all the isotopes of oxygen will get separate out. So that's the technique actually. So that separation will take place in the mass spectrometer, and then there is this detector here. Based on which angle this element or isotope will get separate out, you shift your detector on that position, or you can put at least 16 to 18 different detectors so that you can analyze maximum isotopes or masses at the same time. Okay, so there is this single detector or multi-detector system available in the market. Okay, so for theory, as you can see here, all of this I already discussed. You have this gas sample introducing into the mass plasma here, which is formed by this coil electromagnetic induction, and then you pass these ions through the lenses, or that will accelerate the lenses, that will accelerate the atoms, and then this will go through the magnetic field that will separate out. As you can see that based on the charge per mass ratio, all these 4 lines indicate some particular mass, and that mass represent particular element. So that's 4 different elements you can measure at the same time using 4 different detectors. Okay, that's the story. So what is the different kind of because I told you there are some different features that you can add up, or you can use different instrument available in the market. What are these things available in the market? For example, there is this multi-collector and single collector thing. I told you that the detector can be single, and then detector can be of multi. That is 16 to 18 different detectors. That the advantage with the multi-detector will be you can measure simultaneously multiple elements. Okay, so that's the advantages, but the instrument will be very costly. So some people prefer single collector. So that's mass spectrometer UJ, University of Johannesburg. That's the multi-collector mass spectrometer. And there is this magnetic sector. As I told you that there is this block here. If you can see this steel block, that's the magnetic sector that separate out the spectrum. And then there is this quadruple thing. The quadruple is nothing but it's a 4 aluminum, or some 4 rods, metallic rods that changes their electromagnetic field, and if you pass your matter, the atoms through this quadruple system, that changes continuously, from one charge to different charges, and that will separate out some elements. For example, if you want to just analyze the isotope of oxygen, and you don't want to analyze any matter like silicon, magnesium, or calcium. Okay, So you select that kind of electromagnetic field in the quadrupole, so that it will only allows the oxygen isotopes to enter the mass spectrometer and not the other one. Okay, so that's the quadrupole system. Okay, then there is this collision cell. As I told you, we use the argon gas as a carrier gas for the carrying the matter which is which is dissociated. But sometimes what happens, for example, we wanted to analyze very complicated things like calcium isotopes. So calcium has calcium 40 isotope, calcium 41. So actually the problem is calcium 40 isotope has interference, great interference with the argon 40 because they have similar mass. So what you can do is your carrier gas is argon. You cannot separate, you cannot measure calcium, because if there is interference, you are getting a higher amount of what is actually present in the matter. So what you do is here, what we do is you use a collision cell where or reaction cell where you introduce some gas, for example, oxygen or helium or hydrogen for say, and that for example, helium gas will react with the argon, which is supposed to be interferencing with the calcium 40 isotope. So then this reaction will give you argon, helium, argon, oxygen complex molecules that you can easily separate out by using quadrupole or different techniques, and then you can measure the calcium isotopes. So that's the collision cell. Then there is this time of flight. That's the same thing, like mass spectrometer. But the principle of time of flight is you add you put the different atoms into the time of flight. That's very long tube. Based on their masses, each element will have different speed. Because if I am a bulky man, and if there is some guy which is light inside, which is light in his weight, he can run much faster than me. If I'm very heavy, I have like for say 80 grams of my weight, and then there is this guy 60 kilograms of weight, and then this guy of low mass will have higher speed than the higher mass. So same thing is used in the time of flight. So based on their appearance on the detector, you cut out the signal and you measure one element after one another. Okay. And there is this laser ablation system, because as I told you that there is complex technique of sample preparation. You need to crush your samples and make it into solution. But this sample preparation technique is not easy, because there are a lot of complications. You need to use, for geoscientists, we use very concentrated acids, for example, hydrogen fluoride, hydrogen, chloride, and these things, which is very dangerous acids. So for the laser ablation has put these things away. So what we do is, instead of putting this sample into solution, you put your sample as a solid under the laser. And what laser is doing is, laser is crushing your sample for you. Okay, so you put the laser, and that laser will put a hole into your sample, and that sample will get out from the its original space, and then you can move the sample by using gas to the plasma, and you can analyze. So that's the convenience nowadays came into the market. Okay, so you can use the laser ablation system for analysis. Okay, now the last system I'm going to talk about, the electron microprobe analyzer, which is also similar to secondary electron microscopy, which is most of the people, I think, that you may be using for your images, making images of your biological samples. Sometimes you use cryo-TEM, sometimes you use cryo-SEM. So this is similar to those instruments. So here what we do is we generate this secondary X-rays similar to XRF by using electron beam here, because that's the electron probe. We don't use X-ray beam here. We use electron beam. That electron beam interacts with the sample, and that will give you different spectra. So this is also a wave dispersive method, okay, similar to XRF. So this is non-destructive, rapid, simultaneous analysis, easy for sample preparation, because you are not crushing the sample and making into homogeneous solution. Okay, you are just making thin sections. You take a rock, grind it into thin section. That's it. Okay, and then you can. There is also conveniency here that, for example, that as earlier I told you that there is a watermelon, and if you want to analyze the seed of watermelon, you can analyze the smallest seed. You know, the smallest part of that watermelon as small as 10 microns. Okay, so that small part of that particular matter you can analyze by using electron beam. So detection limits are actually higher. That means that higher means, you know, you cannot go to lowest detection limit like ICPMS. ICPMS measures detects elements up to the parts per billion billion level. Okay, for ICPMS we succeeded up to, for EPMA we succeeded up to 10 ppm. Okay, for particular element. So that's the detection limit, which is, you know, not good compared to ICPMS. So this instrument we can't measure the isotopes. We can measure the elements here, and into that we can't measure all the elements at the same time, because there is limitations. For example, here if you see, as I told you that we use the crystals to separate out our spectra, and each crystal has some limitations, because each crystal cannot cannot disperse all the elements. But for example, there is this tap, L-plate, plate, leaf crystals that will, you know, there is this range of atoms that can it separate out. So for example, for K alpha line, which is, you know, much intense and more easy to measure. So for tap crystal, it measures from, say, you know, for say 7 atomic number from 7 to 14. And when you go higher than more than 80 atomic number, it cannot measure separate out. Okay, so that's the limitations for this EPMA. So moving on to the sample. So these are nice images from South Indian kimberlite diamonds that we studied during my PhD. So in this diamond, for example, every diamond has, some diamonds may have some inclusions. For example, this diamond here has some rock inclusions. For example, here we have olivine, garnet, graphite, sulfite. So if you want to analyze these elements, these inclusions, you can either use Raman spectra. Raman spectra will give you what is the matter, like, you know, you can identify garnet, olivine, or sulfite, but Raman spectra cannot give you what is the composition of that graphite or olivine or sulfide. Okay, there can be a very, you know, bunch of elements that can be there. Okay, so then we can, what you do is here I have shown here you some images. This one is the CL images of this diamond. So you cut the diamond and get CL images, and you will find that there is the diamond is not homogeneous, but it has very small layers. Okay, so these layers represent some impurities in the diamonds, and let's move to the that each zone you can analyze. So these are samples that you can analyze. Like here I have this garnet and CPX, very nice ilmenite grains. And for biological samples, you can do imaging. These are fossils, biological fossils that you can image using the EPMA. That's the secondary electron imaging for the fossils. That's the image for fish. That's the BSc image for tooth here, and that's the bone. Okay, so can you hear me, or am I? Hello?
Yes, sir.
Okay, sorry, because I thought that I just, you know, got disconnected from the system. Okay, so I will just finish in a few minutes. That's the only few slides. I'm not going to show all of the slides. That's end of this session anyway.
Sorry to disturb you. Sorry to
yes.
disturb you, but we have only 10 minutes, so that's why.
Okay, I will finish in 5 minutes. Don't worry. Okay, thank you for you know, okay, letting me know. Okay, so for EPMA, we introduced, this is the atom. We introduced the electron beam here. That electron beam similar to XRF. That dislodge the electrons. That's the characteristic X-ray, secondary X-ray generated because of the jumping of the electron from higher energy level to lower energy level. The same thing, and then you use this interaction. I know for EPMA, if you put any kind of beam onto the sample, that doesn't give you just secondary X-rays. That also gives you secondary electrons. That also gives you cathode luminescence. That the photons get emitted from the lower energy interactions. That also gives you backscattered electrons, because some electrons, they come back from the 90-degree angle. Okay, that doesn't give this doesn't dislodge any electrons, but just come out. Okay, from the surface. So there is the secondary electron, backscatter, X-rays, cathode luminescence, and there is also another thing, auger electron. That's the electron get dislodged from the shell. That auger electron will give you some different information. So this whole bunch of spectrum will give you different information. Like I told you earlier, I showed you diamond images, that is from cathode luminescence. Then there is the secondary X-ray that will give the composition. And then there is the secondary electrons and backscattered electrons that will give you the imaging. Okay, that you can image, you can take the images of that particular sample. Okay, so for theory, same thing. You introduce your electron beam onto the sample, that will generate a whole bunch of different spectrum. You take your, for example, for secondary X-rays into your grating. That grating will disperse this secondary X-rays, and you can measure by using counters. For example, there is this scintillation counter here. So the EPMA parts, there is different parts. For example, this is gun. This is the gun, electron gun. There is this column which contains a lot of magnetics and electromagnetic system things that focus the electron beam onto your sample. That's the sample stage holder, and that's the spectrometer, which has the grating, and the is the detector. So I think I already told you. And for takeaway home message is this is for the students. Mostly for the student, there are two different spectroscopic techniques, WDS and EDS. Secondary X-rays get emitted by any kind of beam that will fall onto the sample. For example, X-rays or electron beam that will generate secondary X-rays, and these secondary X-rays are characteristics of each element. So that's the advantage of the spectroscopic techniques. You can measure the each element by using their characteristic X-rays. Okay, wave dispersive method is more convenient. It's more useful because it measures, it disperses the spectrum in a easy way, and you can measure lots of elements. Okay, so we use for this wave dispersion, we use the Brack law to n lambda is equal to 2d sin theta. For mass spectrometer, we use Q by M ratio. And for XR, for different instrument, for example, XRF, we use measure and trace elements. Icpms, we use trace elements as well as the isotopes. And for EPMA, we use in situ measure and some trace elements. So we cannot measure all the elements by using EPMA. So thank you for this talk, and Juma Mubarak to all of you.
Thank you, sir, for such a wonderful presentation for the spectrophotometric methods in relation to geoscience and the life sciences, and the different techniques on the X-ray technique and ICPMS and EPMA techniques. So
if you have any questions, please ask. Okay.
Yeah, I have one question, sir, to you. Can we use ICPMS technique for the identification of plant metabolite or the structural analysis of plant metabolite?
So that's a good question, actually. The biological samples people use, for example, ICPMS is for measuring the isotopes and the trace element compositions. But you cannot measure the molecule, you cannot measure the structure, you cannot identify the structure. For identification of structure, you need either laser Raman, you may need XRD technique, or there may be like cryo-TEM, that's the transmission electron microscopy that you can use. ICP is for measuring the elements and isotopes. Okay, for biological samples, people use HR ICPMS. That's for example, there is this another technique. You may be aware of, what is that? Chromatography. Okay, so that's the chromatographic techniques that you
we
can.
had done at IIT with the help of you, that we had done at IIT.
Okay, yeah. I remember that you did
Chromatography, GCMS techniques.
HRHC, ICPMS, something like that.
HRLCMS technique that we had done.
Okay.
Yeah.
Okay, great.
Okay. So thank you, sir. I think so.
Okay, thank you for this.
And very much on behalf of our J.A.T Trust, the J.A.T Management, J.A.T Senior College, and all from supporting staff, I'm very much thankful to you that you had given such a wonderful presentation and very useful takeaways to the home from your lecture. Thank you, sir. Thank you very much. Now,
Okay, thank you.
I may call upon Professor
I hope that okay.
Yeah. Professor
I hope that
Talwara
student will
present
learn
you out of
from
thanks.
this. Okay.
Sorry, sir.
I hope that students will learn, take this because all the spectroscopic techniques, whatever you use in biosciences. For example, as I told you that there is always similar kind of principle, whether you use any kind of techniques, TEM, SEM, XRD, or whatever. This principle is very easy. So students should just focus on the principle, and then everything gets simple. Okay, thank you.
Thank you.
Good afternoon to all. It gives me an immense pleasure to deliver the vote of thanks to all the dignitaries assembled here for this one-day national seminar on novel approaches in research methodology. Let me first of all start by giving glory to Almighty Allah for making today's event successful. It's my privilege to propose a vote of thanks to acknowledge the contribution of those who have worked hard to make this event successful. On behalf of Department of Botany, J.A.T Arts, Science and Commerce College, I would like to thank our inspiration, Honorable Alhaj Nihal Ahmed Ansari Sahib, Chairman of J.A.T Trust, for inspiring and motivating us to organize such programs. Now, I would like to place our gratitude to our Honorable Principal, Dr. Muhammad Harun Muhammad Ramzan Sir, and our Academic and Administrative Coordinator, Dr. Salma Abdul Sattar Ma'am, for their generous support and guidance for this event. I would like to extend my sincere thanks for today's resource persons, Dr. Syed Abrar and Dr. Azhar Sheikh, for enlightening our minds with the valuable knowledge. My heartfelt thanks to the organizer of this program, Dr. Yahya Khan, Head of the Department Botany, for organizing such a wonderful program. I would like to pass my gratitude to the IQAC coordinator, Professor Munawar Ahmed, for associating with us to organizing this program. Last but not the least, I would like to thank Naveed sir and Ataur Rahman sir for their efforts and support throughout the program, and my fellow teaching associates and non-teaching staff for their sincere cooperation in making this function successful. At the last, I would like to grant heartful and sincere gratitude to all the participants of the program. Thank you.
Thank you, ma'am. Thank you, sir, Abrar sir. He is also in touch with you, and you are also connected with us in both the section. Thank you, Abrar sir, and thank you, Azhar sir. You are also there for both the sessions, and from a call that you were accepted my invitation. Thank you, sir. Thank you very much.
Thank you very much for giving this opportunity. Thanks a lot. It was nice to interact with Azhar sir as well.
Yes, sir. Thank you.
Great.